Welcome to the fFTD annual seminar. Nice to see you all. You'll hear from me during the day, but I am going to let Nikki, who all you know, just introduce and do a few notices, and then we'll get straight into a couple of talks from Sophie and Liset this morning. Off for lunch, hear about one of our psychology programs and our psychology team, and then hear from some of our members. And then there'll be a bit of time just for questions and other things before the end of the day. So lovely to see you all, love to see you all. It feels like maybe it's the start of summer, but maybe it's not. We'll see, but I'll let Nikki do the start and introductions.
Thank you. Lovely to see you all today. Thanks for coming. I hope it's going to be a really informative day for you. The team's here today that if you need any assistance, there's myself and Emma and Ollie from the RDS team. I'm sure you've been in contact with Emma, and I'm sure more of you will be in contact with Ollie after today. And then we have the research team from John here. We've got Sophie, Amelia and Liset. So welcome to those. And again, if you've got any questions on any of the research, please do spend time sort of talking to them today. Just a bit of general housekeeping. So toilets, they're through the main sort of foyer, and there's men's and ladies' toilets out there. There is an accessible toilet here if anybody needs one. We have not got a fire alarm planned today. So if you do hear anything, there's a door here and there's a door here. So we'll get out as quick as possible. We're actually in a good room for that today, so that's great. As you notice, we are recording, but we are just recording the talks. So it'll just be obviously the introductions, Sophie's talk, Liset's talk, and then Ollie and Sam's after lunch. We've got a members talk, which we will not be recording at all. And we've got Q&A, which we won't be recording either. So please don't feel that anything you say will be on screen and on camera, because it won't be at all. We will be taking notes this afternoon from the talks. We won't be recording, so we will put that into a document with the questions afterwards for you. We'll be doing some online feedback later, so you'll get an email later with a short survey. Please do give your feedback of how you felt the day went, or things that you wished were also incorporated in the day, or things for next year. It's really important to help us shape our services going forward. I hope you like this room. It is the venue that we've been using this year so far. For the bigger meetings, we've been using the room next door, and we've been really happy with it. So hopefully we'll continue to use this. But we will have our very exciting Rare Dementia Support Centre opening next year. Building works will start soon, and we hope it to be open sort of autumn time next year. We'll have plenty of rooms there, which aren't huge big rooms, but rooms possibly this size, which will be able to fit people in. So we'll have some meetings there, and hopefully we'll still be able to have some meetings here. But the centre will be there for people to drop in, have coffee, to have small groups in, to come in after your research appointments and your clinic appointments. So we really hope it's going to be a home from home for you all. Thanks to all of you who managed to do the bookings through Eventbrite, and sorry if we've had to come back and forward to ask for different people's names, but it's really important that we can include everybody's names for obviously health and safety if we do need to get out quickly, but also so we know who's on our membership. And as you all know, RDS has been going for several years now, but it's been growing immensely over the last couple of years. We now have over 6,500 members. I don't know all of your names, but most of them Emma keeps telling me. So I think I'm up to about 6,000 people's names I've remembered at the moment. But it's really lovely that we've got the different groups and we can offer as much support as possible to everybody. We're going to hear a little bit about the peer support groups this afternoon, about the gene carrier group, but also please remember we've got carers groups as well, and we have people we have groups as well, peer support groups for people living with FTD as well. And on top of that, we do offer support, one-to-one support for all of our members, and people can contact us anytime using the email address here. We're just changing our support over to more like virtual support clinics. So it means that when people do come to us requesting a support call or a Zoom call, Emma can slot them into a clinic appointment, and we can do that within 2 weeks. So you can take that back to your GP, see if they can beat that. We have the lovely Shivani here. She's sat at the back, who's from the National Brain Appeal. So if you have any thoughts on fundraising, you want to run a marathon, you want to sign your husband up to run a marathon, or have a bake sale, do a concert, anything like that, Shivani is full of lots of ideas to help. And we are really so appreciative to the National Brain Appeal, who allows us to put these days on. So thank you, Shivani and team. And we'll just start then. This is the agenda for today. We've got some fantastic talks. So Sophie's going to talk first of all, then we're going to go to Lisette. We'll have nice break for lunch, and at lunchtime it's really that important time for you to speak to each other. It's only once a year that we manage to get together in person with this, so please spend that time talking to each other. And then in the afternoon, Sam and Ollie will start us off with the online IWARF project. We'll hear from our members, and then, as John said, we'll finish off with update on clinical trials and a Q&A session. So I'm going to hand over to Sophie.
Thank you. Great, so it's lovely to see you all here today. For those of you who don't know me, I'm Sophie Goldsmith, and I'm currently working in the Ray lab at UCL. I'm going to talk to you today about my PhD, and my PhD is the Familial Frontal Temporal Dementia PhD studentship funded by the National Brain Appeal. So, a quick introduction for those of you who have not met me before. I did my undergrad and my master's at Bristol, but then back in 2021, I moved to London and I started working with John and his team, running the UCL part of the GEMFI study, which for those of you who don't know is the Genetic Frontal Temporal Dementia Initiative. And that's an observational study where people come in and they have a brain scan and a blood test, etc. So I did that for just over 2 years. And more recently, I've moved to the lab, so quite a big change for me. So back in February, I moved over and started my PhD in the labs just around the corner here. And so today, I'm going to talk to you about what are mini brains and how do we make them? How can we use these to study disease? And then specifically, what's my PhD and how we're using them to study frontal temporal dementia? So, as you probably already gathered, my PhD project focuses around building mini brains. And so here's a picture of them. And so just to give you a bit of context as I talk through it throughout the talk, but they're about 2 weeks old here. And I thought I don't know, I think they look quite pretty, but maybe actually they probably don't look that nice. But so how do we make these in the lab? So going back quite a few steps, what we do is we take a small skin sample from the top of your arm, and we take these to the lab, and what we can do is we can grow them on small dishes. And what we need to do is to be able to use these to then make the mini brains, we need to almost switch these cells back to what they once were before they became skin cells. So what we do is we reprogram them, but what I mean by that is just give them some food with different chemicals. And what we can do is then turn them into pluripotent stem cells. And all you need to know is these stem cells are the ones that then eventually return into other cells in your body. So that's a bit complicated, but all I'm saying is that we can turn skin cells back into these stem cells to then make the brain cells. So how do we go from the stem cells to the mini brains? Because that seems like quite a big jump. But actually, there's been a lot of advancements in the technology recently, and it doesn't actually take that long in the lab, and it's not super complicated, which is to my surprise, because I've never done this before until February, and I was quite surprised with how straightforward it was. But you can see here, so you can see here that you've got what we call a colony, but it just means a group of those stem cells that I was talking about. And what we do is we grow these up for a couple of weeks, and then we feed them with what we call media, but what that is just the red stuff in the bottom there. And it's essentially food. And what it has is lots of chemicals that mimic what is happening during development. So when in the embryo. And what this does is it basically switches on different genes to make the cells become brain cells. And that sounds all quite complicated, but I'll just tell you a bit about why this is helpful. So if you remember back to the diagram where I showed you that you can take skin samples, but you can take skin samples from people with mutations and people without mutations. And what this means is that you can then grow up those organoids or mini brains, and they hopefully will be a really good representation of what is happening in those people. And that's really helpful because then we can see if there's a difference between the people with the mutation and the people without the mutation. So I just thought I'd show you a very quick video just so you can see what they look like in real life. So those dishes are about that size for context. And they grow up and you keep them in what we call culture, but that just means feeding them and keeping them warm essentially for about 3 months. And so these are they grow to about the size of a pea, so not massive. And of course you can see they don't actually look like brains themselves. I think mini brains is quite an ambitious name for them. They're a collection of cells that are representative of just some of the cells in the brain. And so to take a step back, just to give some context for my project, I just wanted to talk to you about the kind of cells that we find in the brain. And just a caveat that this is massively simplified. There are way more cells in the brain than the ones I'm talking about, but I'm just focusing on the ones that are relevant to my project. So firstly, you've got the neurons, and these are the cells that take in information from your surroundings, and they help to produce all the outputs that we do. So speaking, walking, thinking, eating, all of those outputs. They're very, very important for our everyday functioning. And then you've got what we term is the immune system of the brain. And these are the astrocytes and the microglia. But essentially what they do is they move around in the space between the neurons, and they make sure that the environment is optimum for the neurons to do their job. And that's very important so that we can have all those outputs, and so we can speak and talk and everything, and it all functions properly. So what can we make? So as I said, we're making the organoids, the mini brains, and these contain both the neuronal cells and the astrocyte cells. But we can also make, and when I say make, I just mean we take those skin cells and we give them slightly different food. We can make the microglia as well. And why this is very important that we can do both of these, because if we just had the mini brains, it wouldn't be a very good representation of what is going on in the actual brain. But if we can take these and combine them, then hopefully it's a better model. So what can we do with these models? So as I said, you grow them up for a few months. And then what you do is what we call harvest them, but by that I just mean take them from their food and from the warmth, and we can cut them up and stain them, which feels a bit bad after growing them for so long that I just take them like that, but stain them for different markers so that, like in the picture before, so you can, there we've stained them with a fluorescent dye essentially that just tells us that these are the cell types that we're hoping they are. You can then homogenize them. And what I mean by that is literally blend them up. So you get a pestle and mortar and mash them up, which again seems a bit aggressive. And that means you can measure different proteins. And finally, you can also extract the DNA and RNA. But ultimately, the most important thing to know is that this means that we can answer the question, is there any difference between those mini brains that we've made from people without the mutations versus mini brains we've made from people with the mutations? And so we can see if there's anything going on that's different in these models. And so how am I using these mini brains to study familial frontal temporal dementia? And so my project specifically focuses on progranulin-associated frontal temporal dementia. And what we know already is that when you take a blood sample from people with and without the mutation, you can see on the graph, you can see in the blue, you can see that these people don't have the mutation and they have a normal level of progranulin. Whereas if you've got the mutation, you can see that they have around half the amount of the usual level of progranulin. So that we know. We also know it happens in blood, but we know roughly what happens in the brain. And we know that when you have half the amount of the progranulin levels that are usually there, you see this buildup of this TDP43 protein. And all I mean by that is there's a protein in the cells that clumps together and basically clogs up the neurons and leads to cell death. So that's ultimately why it leads to frontotemporal dementia. But why does this happen? Well, we don't know. We actually don't really have a very good understanding of why the lower levels of progranulin leads to frontotemporal dementia. So that kind of brings me on to my project. And this is where it might get a bit heavy in the science, so just bear with me. But one theory is to do with what we call the lysosome. And so in all of those cell types, but actually in all cell types across our body, we have these things called lysosomes, and they're basically bubbles filled with enzymes. And what they do is they go round and they clear up all the rubbish in our cells. So they're basically rubbish collectors. They eat everything up and they make sure, therefore, that the environment that the cells are working in are rubbish-free. And we know that in progranulin-associated frontotemporal dementia, that these lysosomes aren't functioning how they're supposed to be functioning. And one of the ways we have particularly good evidence that this might be what is underlying it is, so when you have 100% of the progranulin, we see that in the control cases. When you have half the amount of progranulin, that's what leads to frontotemporal dementia. And in very rare cases, you have absolutely no progranulin at all. And this leads to a set of conditions called neuronal ceroid lipofuscinoses. And these are lysosomal storage disorders. So these are disorders where the lysosome is not working at all. And basically all I need you to take from that is that progranulin is very important. And we know that it's very important to lysosomal functioning. And so the rubbish collectors aren't working in this condition. And so to complicate matters further, just to give some more context of my project, we know that having half the amount of progranulin, particularly in the microglia, so just to remind you, there's the cells that are going around making sure that the environment is correct, leads to particularly bad lysosomal functioning. And so we've shown that in lots of different studies before. And so this is the main slide, is that this is what we think is the mechanism underlying this disease. So we think that reduction in progranulin, particularly in the microglia, leads to changes in the lysosomes, so the garbage collectors aren't working properly. And that leads to the buildup of this protein in the neurons and subsequent cell death. And I know that's all a bit of a mouthful, but my project, just in one sentence, is: do changes in lysosome function in the microglia lead to buildup of proteins in the neurons and subsequent neurodegeneration? And so what I'm doing is I'm going to take those mini brains and the microglia. So at the beginning, I showed you that you can combine them. And what I'm going to do is a sort of mix match approach. I'm hoping this diagram kind of explains. But so taking the mini brains from people without the mutation, and as well as the microglia from people without the mutation, and then doing the same thing but in reverse, so having mini brains from people with the mutation and microglia from people with the mutation. So we think there will be a good model of what is happening in the brains of those people. And then what we can do is slightly complicated. We can kind of mix match them, and so have brains with the mutation and microglia without. And that just basically means that we can kind of see what is happening in each cell type and kind of get to the bottom of what might be actually happening in this disease. So what can we measure? So we can measure the levels of progranulin, similar to what we can do in the blood test that I showed at the start. And that's just basically reassuring us that these mini brains are very similar to what's happening in the real brain. We can measure the amount of the TDP43 protein, and that's just the protein that clumps together in the neurons that I was talking about. And again, it's just another way of us confirming that this mini brain is a model of what's happening in the real brain. And finally, we can measure disruptions to any cellular processes like the lysosome, so we can see if those processes are going wrong. And so what we're expecting, well, we're hoping, it's a process, but we're hoping that we'll see that these mini brains have decreased progranulin levels in the mini brains with the mutations, again, meaning that it would be a good model. We think we'll see that there'll be those clumps of the proteins, and we also think there'll be abnormal lysosomal function. But all of that to say, how could this bring us closer to potential drug treatments? And essentially what I'm trying to do is just make a model of what is happening in real life in the dish so that we can see what's happening and then hopefully try and increase the progranulin levels via lots of different methods, and then hopefully rescue any changes that have been seen previously, the ones I've just described. And we're hoping, this is a long way off because I've just started, but we're hoping that this will then be a good model for drug testing going forward, and hopefully a better model than the mouse models that we currently have. And so if you didn't take any of that in, because I understand it's quite a lot of sciencey stuff, the main messages that I want you to take away is that we can make mini brains in the lab from skin cells, which I just think is like phenomenal. As I said, I just started in Feb, so I was at the same stage as you now, where I was equally as surprised as that this could happen. We can understand that you can use this to understand the biological mechanisms underlying FTD, and hopefully use this model as a model for drug testing going forward. And my project is focused on progranulin associated frontotemporal dementia, but we can hopefully use this model in lots of different scenarios. And as I said, I just started in Feb, so I don't have any data to show, but hopefully at some point in the future, I'll have more data and hopefully some exciting data to share. But big thank you to all of my supervisors and the Ray Lab, and of course participants who've donated any skin cells for this research. But if anyone has any questions, then do let me know. If you want to catch me in the break, then let me know.
Great. Well, Sophie's you'll be around and you can ask some other questions about things. So thank you. I will introduce Liset de Boer, who is an FTD researcher who's been visiting us at UCL for the last 6 months. Is in the middle of her PhD with one of our really very close collaborating groups in Erasmus Medical Center in Rotterdam, and in the worldwide study of familial FTD. I always like to think that we have obviously have a lot of families who we worked with over the years, but not as many as in Erasmus. So currently in the GEMFI study, UCL is number 2 in terms of the number of visits. We've had about nearly 600 people, 600 visits in the GEMFI study over the last 10 years. But at Erasmus, they've had over 1,000 and have lots of families, and particularly progranulin families, who John Van Sweeten, who runs that group and has been running that group for many years, has met over the years. And so they have a big cohort of people, of families that they have been involved in their research for a very long time. But as part of our very nice collaboration, we often have visitors over here for a little bit of time. And Liset's expertise is in cognition and thinking, and she's come here and been thinking a little bit about how we use eye tracking. Some of you will have kindly helped with those studies, but Liset will tell you a bit more about why we use eye tracking and what we do.
Thank you, John, for the nice introduction. So for the ones who have already heard me speak this morning or are hearing me now, I'm not from England. I'm from the Netherlands, as John said, but I'm trying to do my best to make everything as clear as possible for you. But just let me know if anything is unclear or if you want me to explain a bit further. So as John said, I am from Rotterdam. Well, I'm not from Rotterdam, actually. I was born in the northern part of the Netherlands, in a very farm-like village neighborhood. They didn't have lots of universities there. I had to cycle for 1 hour to go to high school. So that's why I decided to move to the southern part of the Netherlands to study there and to work there as well. So I graduated as a neuropsychologist. I graduated at the University of Utrecht, which is a lovely city. The name Utrecht is a very Dutch name for a city. It's very close to Amsterdam. It's actually a bit of the tiny Amsterdam, we like to call it. But it has an amazing university. And after graduating, I was asked to start my PhD in Rotterdam, which is a larger city. I'm not living in Rotterdam. I live in The Hague myself, which is a city next to the beach. So it's very nice for after work to just hang out on the beach and just relax. The commute to Rotterdam is approximately 45 minutes. So I was quite shocked when I decided to live in London that my commute will be around 1 hour. Well, in the Netherlands, you can just travel for 45 minutes from one city to the other. It takes 1 hour to travel within London. So that was quite a cultural shock for me. As John also already said, in Rotterdam, we are working on the GenFi study as well. We are a very big site, and we always love to collaborate with UCL in London. That's what we really, really like. In Rotterdam, I also see all the participants myself. So I'm doing all the intakes. I call their family members or their partners for more information. I'm doing the neuropsychological testing. So I'm seeing them for like hours in one day. Sometimes I'm also doing the MRI scan. So I'm really thankful to stand here in front of you today to also hear a bit more about your experiences, because I've been talking with the participants in the Netherlands for a long time, but I'm also really curious about how you feel, and if you have any questions, just please let me know. But let's go to my talk, because that's why we're here, right? One of my projects during my PhD is to study whether we can use eye tracking to measure our brain functioning. And I think nowadays we're really making the shift towards more innovative digital measurements, which can be quite scary as well. But I think it's a really, really great step forward. For more like innovative measures. So today I would like to talk about, first I want to introduce what are cognitive functions. As a neuropsychologist, we are really focused on these cognitive functions and why are they so important in dementia. Then I would like to introduce to you what eye tracking exactly is, how it works. I'm not going in too much detail over there. But I do want to introduce to you how eye tracking normally works and what we do with it. And then I would like to talk about how we use eye tracking in healthcare. Eye tracking has been used for lots of years in other sectors as well. For example, in marketing, they use it for website development, but using it in healthcare is quite new. And then I would like to discuss with you as well what the advantages are of eye tracking, but maybe also what the disadvantages are of eye tracking, and how we can use this and implement this in the future. So let's first start about what are we measuring. So in our brain, we have these different brain areas that all have different functions, which we call cognitive functions. And these help us to think, to react to the world, to make sure that we can do whatever we want, and also to make decisions. We can separate the cognitive functions into memory. So, for example, recognizing faces, recognizing places, remembering what you've eaten yesterday. I always forget what I eat yesterday, but those things are really like your memory, what we call memory. And you can divide memory also in specific types of memory, but let's just talk about memory now. Memory is mostly located in the sides of the brain, what we call the temporal lobes, specifically the hippocampus. It's very important to have memory. Otherwise, we wouldn't be able to sit here because we would have forgotten our appointment of being here. Then in the same area, we do have our language. So that's how we speak. So also motor-wise, so how we use our voice to speak, but also remembering words. This is what we call semantics. And also, learning different languages also has a lot to do with memory and the language area in our brain. Then we have our attention, which you all have very well, because you're all here listening to me, having attention to me. It's mostly in the frontal side of the brain. In that area, there's also problem solving. So how we solve problems, how we can be flexible, how we can sometimes inhibit ourselves, not speak to everyone about what we think. These are all in the frontal areas of the brain. And then we also have processing speed, which is the speed on how we process things. It's literally the speed how we can, for example, read, watch television, hear someone speak. We all need to process it directly. And then lastly, which is one of the cognitive functions I'm mostly studying right now because it's also often affected in frontotemporal dementia, is social cognition. This is a relatively new domain. And this is about how people behave in social situations. And that's, you can also separate within how do people recognize other people's emotion, how people can have like empathy for other people, but also how they behave in social situations. For example, here, we all know that you need to be quiet. That's like nice for me to, while presenting. That also has to do with social situation. You can all empathize with me that I'm here, so you will be quiet. Some of these cognitive functionings are impaired in frontotemporal dementia, but also in other types of dementia. So, for example, in Alzheimer's disease, we see that memory is often the first thing to be impaired, affected. In frontotemporal dementia, we also do see memory, but I think at the beginning it's more people's behavior. So social cognition is a very important one for frontotemporal dementia, but also language. We see that some people often have difficulty with understanding people or with expressing themselves, but also the attention is a bit more reduced. So these cognitive functions are very important to study when someone comes in with complaints. We can create like a cognitive profile of all these domains and exactly tell someone what is going wrong in the brain. Then what is eye tracking? So we can study these cognitive functions that I showed you with eye tracking as well. And eye tracking is, it's like a camera in front of a screen that detects eye movements. In most cases, people are presented with images or stimuli on a computer screen. In front of the computer screen, there's the eye tracker, and we measure the eye movements while doing something on the screen or while looking on the screen. And then we have very beautiful software to analyze this data and to also reveal patterns in how people look. So don't want to go into this in too much detail, but this is how eye tracking works. So here we have someone, I think, playing a chess game on a computer, and in front of it, there's a kind of a big eye tracker. There is a camera on the eye tracker and what we call an illuminator. And the camera measures the reflection that is on the corneal side. So we identify the pupil, that's the blue around, the eye tracker detects that, but it also looks at the reflection of the light to exactly know where someone is looking. So why are we using this in healthcare? I think it's a great way to create a very detailed picture on cognitive functioning, so how someone thinks. It is easy to administer because, well, especially for us, it's easy to administer because we don't have to spend hours on neuropsychological testing, but we can just give put the eye tracker in front of someone and explain some tasks. And it's most of the time it's comfortable. Well, testing, of course, is never really comfortable, but I think it's more comfortable in a way that the classical neuropsychological testing, so creating a cognitive profile, can take hours. And with eye tracking, it's faster. So it will take less long. And people are also, it's all about the eye movement. So it's not about you having to give like verbal responses. So it's it takes a bit less energy. And in the future, we would like to use eye tracking to help getting a diagnosis. So we can create eye tracking profiles for specific types of dementia, which can help us that if we see, for example, someone who is very impaired in an attention task, that we could say, okay, this is probably more FTD related. And if we see eye tracking impairments in, for example, a memory task, we can say, well, maybe this is more going to Alzheimer's disease. And it would also be very interesting to use this to monitor how people progress in their disease. What we know is that eye movements, like the basic eye movements, are often not impaired in early stages, but will, in a later stage of dementia, will also be more impaired. So we can, as I said, we can measure the thinking with eye tracking. Sorry, the layout of my presentation has a bit shifted, but here it says measuring thinking with eye tracking. So at the Dementia Research Center, where I work right now, we have a very tiny eye tracker. It's called the Toby. It is this is the eye tracker. It's like this big. It's a very small eye tracker, and we can stick it on a tablet. And on the tablet, we show different tests that measure our cognitive functioning. This Toby is compared to the other like big eye trackers that they often use in, for example, marketing. It's relatively cheaper, which also makes it a bit less sensitive. But with the research we have done, it is sensitive enough, and we do really see differences in performance in people who have, for example, genetic FTD and are in their early phases of the disease compared to healthy controls who do not have a family history of FTD. We can measure, for example, attention, as I said, and how we do this is we let you watch a cross for 10 seconds without blinking. You really need to focus on that. Myself, I am not able to do it all the time. Like without blinking, it's very, very, very hard, and it makes you cry 50% of the time. But it really is a very specific measure to get a bit of insight in how attention works in someone. Then we can also measure problem solving, and then especially mental flexibility and anticipation. So, for example, how well you are able to shift within of shifts in one assignment into another. I'm not sure who of you have done some neuropsychology testing, but we have a test where people have to draw lines from 1 to A, 2 to B, 3 to C. I see some nodding. Yeah, it's a very, very, very classical psychology test. We can also do that one on the eye tracker, and we can exactly see how people look from 1 to A, 2 to B, or for example, maybe they first look from 1 to 2 and then are like, oh, I forgot, I need to go back to A. And we can really see what is going on in the brain, which you don't really see on pen and paper because we are not able to measure how you look and how you think about it. But I think it's way more specific with an eye tracker. And then lastly, we can also measure social cognition, and then especially the recognition of emotions. And that's, again, that's one of my projects that I'm working on at the Dementia Research Center. So, for example, here we see a very happy face. I hope people will leave this room with this face, not with these. We see a scared face representing fear, and someone looking really disgusted. Maybe she's seen like a very rotten apple somewhere laying on the ground. And with these images, if we present this in front of an eye tracker, we can really see where someone is looking. We do know that with recognizing emotions and seeing emotions, people often look, for example, to the eyes, to the mouth, because that's the parts of the face that really gives us the information of the emotion. And we can really see this with eye tracking, like what are the patterns that people are looking to faces? Maybe some of you might have like a family member with frontotemporal dementia, and often this emotion recognition is affected. We often do see that if we study people with frontotemporal dementia with eye tracking, we see that they often look at the middle of the face, at the nose. So they do not really grasp the eyes and the mouth and the important information of the face while seeing any emotion. So that's definitely a very important aspect regarding emotion recognition in frontotemporal dementia. My project, like my test, so I developed my own test on the eye tracker. It's a bit different from only showing these emotions. It is actually about looking at the emotion and then linking one of these images with the emotion on the face. So what I do is I first present 4 images, then I show you the face with the emotion, and then I show the 4 images again with the emotion on the face. And what I expect that will happen is without seeing the face, people look approximately the same amount of time to the images. So that's what you see in this, we call it a heat map. The green is where people are looking, and if it's red, people are really focusing a lot on these images. And then after presenting the face, we see that the time spent looking on these images is more reduced, and the image that really matches the face, so this, you don't really see it, but it's a very scary man who is probably scared of the gun. So you see that people are looking longer to the gun. So that, yeah, it is a measure of social cognition, as people really need to identify the emotion that's presented on the face, but it's also linking emotion with an image that is presented on the screen. I don't want to show this slide right now because I want to ask you first. After hearing about this, what do you think are advantages of using eye tracking in cognitive testing and in, for example, using it for diagnosis or treatment monitoring? And what are disadvantages? I actually would really like to hear from you. Think about it for 1 minute. Yes.
The advantage would be with a small tracker, you could do it at home, so you don't have to come to a medical environment.
Absolutely, yes, yes. That's really one of the big advantages, that you can do it from home. Any other?
With the results, doing something from home, not in a controlled environment.
Yeah, so with the eye tracker, it is especially important to be in a quiet room, which is quite dark. So that's actually one of the disadvantages, because you're not really able to control it. I think I'll see it in a way that someone is visiting someone with the eye tracker, making sure the environment is more controlled. But I think the eye tracker is quite capable of controlling for not controlled environments. Yeah, but that's definitely one of the disadvantages as well. Any other ideas about eye tracking, or how do you feel about using eye tracking? If you're, for example, doing a study visit or coming for diagnosis and you're presented with an eye tracker, how would you feel?
I think a disadvantage would be sometimes the attention span person with it, because they don't always, you can't always get them to do what you want them to do.
Yeah.
So, to get them to sit still for a period of time may not be successful.
Yeah, absolutely. Yeah. So one advantage is that it's more comfortable because it doesn't really require a lot of instructions and verbal responses. But on the other hand, that also makes it more, I can't say boring, of course, but it makes it more like you really need to focus on that. For example, our study protocol, we also ask people to look at images. I think it's like there are 3 different tests, I think a total of 20 minutes. So imagine having to look at images for 20 minutes long. That can be quite exhausting. Yeah. So what I wrote down myself, these were the advantages that I found. It's very precise data. So normally we're not really able to grasp the subtle things during our neuropsychology testing. We're human, we can do a lot, but the eye tracking can create very precise data and very precise patterns. So it will be more sensitive in an earlier phase of the disease. On the other hand, I think the sitting with a neuropsychologist doing the testing also has its advantages. It's more human, I think, instead of sitting in front of an eye tracker the entire time. It is non-invasive, and which means that it's comfortable, but also it doesn't require lots of instructions, and it also doesn't require a lot of responses. And that's what we call that it's non-invasive. Some of our classical tests can be quite invasive. If we ask you to name as many animals as possible for 1 minute, you're like, oh, how am I going to do this? And we get immediate insight in how someone is performing. So the software really helps us to create really good patterns, like the heat maps that I showed you. It's just one click, and then it creates a heat map for you. So we have immediate insights, while the classical neuropsychological testing can take around like a few days to really calculate all the scores and create a really profile. Well, we have the portable eye trackers now, which makes it more accessible. So we could use it in our lab, or we can also move it to the hospital. We can move it to someone's home, so it's more accessible. In the future, we could also combine it with other tools like MRI, or even with virtual reality. So one of the projects in Rotterdam, where my colleague, it's one of my colleagues' projects, she's using virtual reality with eye tracking to create social situations. And so it really gives you the feeling that you're more like in an environment and not really in a test setting. And we could also eventually use it on smartphones, where the camera of your smartphone is actually looking at your eye and blink movements. So I think it's really interesting, but also ethically is going to be a big issue. But again, yeah, it's quite promising as well to use it on your smartphone as well. So that was my presentation about eye tracking. I hope it was clear for you. And if you have any questions, let me. Yeah, I think we still have a few minutes. Otherwise, just meet me during lunch.
Great. Well, I feel like time for lunch. I'm right on time. So we've got an hour for lunch, and then back in here for 1, and lunch is back where we had coffee. All right.
We're going to talk to you this afternoon about, well, what you can see on the screen. It's called the Improving Wellbeing in At-Risk fFTD project, or IWARF for short. And what we're going to do today, if this arrow will behave, he says, told you I was an app. There we go. So that's our logo. That's the name of the project. And our structure today is hopefully we're going to do as little talking as possible and get as much room for you guys to give your input. So I'm going to do a little outline of the course for us all. And then Sam's going to do a little presentation of some of your feedback from people like yourselves who've been through the program already. And then what we're going to do is we're going to be very cheeky and use you as free advice, really. We're going to get what you think about it based on what we've shown you, so that when we do roll it out for full trial later this year, it reflects what you want to see and can be as helpful as possible. So with that all being said, I'm just going to start with this. So it's a self-paced online course, and our goal is to promote well-being for people affected by fFTD. And by that, we mean people who are gene negative, gene positive, or don't know their status. So it's currently not being extended to caregivers, but it's specifically for people with the gene in their family. It's split into 8 online modules. We cover 6 main kind of topic areas, and there are 3 check-in sessions throughout with, at the moment, me or Sam. So how those would work is we would talk about how you found the material so far, give you an opportunity to consolidate everything you've learned, ask questions about anything that wasn't clear, have a little practice of bits and bobs. So that's what it is, and it addresses kind of some of the big questions that arise in connection with living at risk or living. When we talk about living at risk for this presentation, by the way, we will include people who know their status, whether for or against, because we know, don't we, that it doesn't stop when you know your status, even if that status is negative. There will be people around you who you are supporting who may not know themselves, and that risk is still a very present factor in your lives. So we're talking about rumination about risk, how we can help ourselves kind of deal with that process and the uncertainty attached with yours and your loved one's status as well. How we can help ourselves live our lives to the full. Very punchy phrase that. We're going to try and explain that more in the course so that it's more manageable and makes sense, really, rather than just a buzzword. And a big part of doing that is connecting with our values. And that's what a lot of the course focuses on, helping you identify and then live according to those. And more, apparently, with an exclamation mark at the bottom. So there you go. There is a therapy model that underlies it all as well, which is called acceptance and commitment therapy. A lot of you will have come into contact with some of these ideas in the kind of moniker of mindfulness. So that's how some of you will be familiar with some of these concepts. And there's kind of, as you can see, there's this little pentagon. And I won't do you the disservice of reading everything that's on the screen, but essentially it's about understanding our values, connecting with them, understanding that they sometimes come into conflict, and helping us manage those difficult periods where we're trying to choose between two values and helping to connect to them as often and as completely as possible. And then the other kind of core part of it is this understanding that we exist kind of in this kind of permanent observer state, and our emotions and thoughts and feelings are fleeting. They come and go. It sometimes feels like they persist and drag and aren't going to go anywhere, but they always do. They always change. And it's this idea of trying to help ourselves understand that the way that we feel in a given moment is able to change, and that something new will follow it, and trying to take that self as the observer stance, as we call it. So that's kind of the core bit underlying it. And this is what the structure looks like. So this is how it looks so far. And again, when I say we want the feedback on everything, I really do. So it's obviously hard to tell from a little presentation, but this is what it looks like for now. And we'll be talking with you and with people like you to continue to hone that down to get it in its best form possible. But we've got these kind of modules, which are the numbered bits. And within them, we've got different videos, worksheets, resources, all sorts. And then we've got the check-ins. And as you can see, this middle bit splits. So if you know your status and you know that you're a non-carrier, not a non-character, you go down a slightly different stream, and you kind of will talk about survivor guilt and anger and the kind of challenges that occur specifically through getting that known negative status. And then if you know you're positive or you don't know your status, you go down a different work stream. And as I said, there'll be professionals throughout at each of those check-in points to help you kind of digest what you've been feeling. You know, tell us it's working or not working, and if it's not working, why isn't it working, and all of that kind of thing. So I think that's me shutting up for a second now and Sam taking over. But hopefully that gives you a little grounding in what it is we're doing.
Yeah, thanks, Ollie. And yeah, just to say those check-ins are to have been on the side of it that we are. It's really interesting. Some of the things that aren't working lead to the best conversations, actually. So if someone says this technique doesn't fit me, that's actually like always been really helpful to us. And I think that yeah, I've really loved that part of the program of sort of sitting with people and problem solving and saying, yeah, this isn't working, and that's helpful. We can move, we can play with these ideas. We want to encourage people to play with them as much as possible. And just generally, yeah, feedback from people that have been in the intervention, either during it or post, has been really, really helpful to us. So everyone, we're in the feasibility stage at the moment, which means that we're trying it out with people. And we're collecting feedback while they're doing it, but also after every time someone's been through the program, we're conducting an interview with them and just asking them some questions about how they found it. And that's been an incredibly helpful process. And we're in the process of, we've recorded those interviews and we're analysing them at the moment. So we're trying to sort of pull out the most important themes and suggestions for improvements that we can. We're in quite an early stage of that, which is quite exciting. So at the moment, this is after sort of looking at only, I think, 4 interviews we've looked at to pull some of these suggestions out, but they've been, or as you can see, there's already like a lot there. So some of the things that we found are that the acceptance content of the intervention, so that sort of idea of mindfulness and being with your feelings, and rather than sort of trying to fight always what you're feeling in the moment, to sort of really sit with what it means to incorporate difficult feelings, anger, all of these things. That people have been finding that a particularly helpful part of the intervention. We've also had some really helpful feedback on the check-ins themselves. So obviously, the way that the course is structured, people have their sort of materials and a bit where they're sort of learning these techniques and finding out things, and then sometimes sort of try them out. And then we have our check-ins where we sort of go over the materials again. That's quite a lot to ask someone to do, I think. You know, it's work really, or it's stuff to do in between. And so we've had some really helpful feedback about the length of the break between the check-ins, particularly when people are juggling work lives or all the many things that can get in the way, to make sure that people have enough time to try these things out and really sort of play with them. We've had some helpful feedback about potentially some of the techniques within the intervention being helpful for many different people with many different connections to FTD, but also just friends and things like that. So a general sort of message to maybe extend recruitment to other people as well, other than people who are at risk. One thing that's come through really strongly is the idea of a refresher booklet. So at the moment, as the intervention stands, it's lots of videos and sort of this content, but people sort of want to have something to take away from the intervention as well. So here's a sort of book of almost like a toolkit of things that you can sort of try, and you can whip out this sort of booklet with some sort of shorthand ideas in there. Some people have talked about how helpful the check-in sessions are for helping them sort of know when to sort of implement or deal with the materials. And so some kind of a notification post check-in to sort of say, right, you've moved from this stage of the intervention to the next one. Some people have mentioned that might be a helpful way to change the intervention. And then the final one, which has been, it's quite an interesting mixed piece of research, mixed sort of finding, which is that the way the intervention works is that it kind of does just throw lots and lots of techniques at people. And as you said, some things are going to speak to some people, and some things are going to speak to other people. And so there's been some positive feedback on that, sort of suggesting like, even if some of this material doesn't fit me, I can see how it might help someone else. And so that's a good thing, but that it would be helpful. I think people first starting the intervention weren't quite so sure that they could sort of pick and choose and sort of decide, actually, this isn't for me. And so some sort of information up front that says, this is about building a tool belt, but your tool belt might look very different to someone else's. And so something to sort of hammer home that message that you can sort of make it work for you. So those have been some really helpful findings.
That's all we've got time for today. Unless there is any other very general, and I don't actually know if I'm over time or not. I should have timed this. I'm over time. No more time for questions. Linking to this slide, if you have any thoughts or questions or anything relevant to this kind of stuff, then please email this address here, and we'd be very happy to hear from you and be in touch with you, whether it's to participate in the trial when it comes up or to kind of give your thoughts and opinions or whatever it is. Please do note that down and send us an email if you're interested.
Massively appreciated.
Yeah,
including everything today as well. Thank you so much. Yeah.